DNA structure. The DNA is made up of two complementary polynucleotides, which means pairing pair with pyrimidine. Each polynucleotide strand has an alternating phosphate groups and sugars on the backbone. The DNA double helix is anti-parallel, which means that each end of the DNA molecule containing the 3' end of one strand and the 5' end of the other strand. The phosphate group is on the 5' carbon and the OH group is on the 3' carbon. Hydrogen bonds link each complementary base pair. A and T share 2 hydrogen bonds, C and G share 3 hydrogen bonds. Now, compacting of DNA. Compacting of DNA happens when our DNA wraps around histones protein, and DNA supercoiling forms due to that, but enzymes such as gyrase controls the amount of supercoiling. Semi-conservative replication. In semi-conservative replication, each newly synthesized DNA molecule is composed of one strand from the original DNA molecule and one new strand. The replication starts at a specific nucleotide sequence called the origin of replication. Helicase enzyme binds to this site and breaks the hydrogen bonds, and the replication bubble will be formed. Single-strand binding proteins will help to stabilize the newly unwound single strands. Topoisomerase II will help to relieve strain on the double helix sections ahead of the replication forks. Primers join to the 3' and 5' end. DNA polymerase III synthesized a new strand from the parent strand by attaching new nucleotides to the 3' hydroxyl end in the 5' to 3' direction in the replication fork. A lagging strand and a leading strand will be formed. DNA polymerase then removes the primers and fills in the space by extending the neighboring DNA fragments, which are the Okazaki fragments that are joined by the DNA ligase enzyme in the lagging strand. Then the DNA returns back to its stable double helical structure. We can form different proteins from our DNA by protein synthesis. Two steps in protein synthesis are transcription, which is the formation of mRNA, and translation, which is the formation of the protein from the mRNA and tRNA.
DNA mutation is a permanent change in the nucleotide sequence of a cell's DNA. Mutations can affect our protein synthesis. There are two categories for mutation: single gene mutation and chromosomal mutation. Single gene mutation is mutation that occurs in one gene, while chromosomal mutation occurs in the chromosome. Under single gene mutation is silent mutation, which does not change the amino acid sequence; missense mutation, which changes the amino acid sequence; nonsense mutation makes protein short by introducing a stop codon; point mutation, which includes deletion, insertion, or substitution of nucleotide; frame shift mutation, which is the insertion or deletion of nucleotide. And for chromosomal mutation, it includes duplication, inversion, reciprocal translocation, and deletion.
Gene cloning is a common practice in molecular biology labs that is used by researchers to create copies of a particular gene for downstream applications, such as sequencing, mutagenesis, genotyping, or heterologous expression of a protein. Genetic engineering is the process of specifically altering the genetic makeup of a particular organism. This can be done by making changes to the sequence of the DNA, such as introducing a mutation into the gene. Genetic engineering process can be used to make genetically modified organisms. Gel electrophoresis is a lab method used to separate mixtures of DNA, RNA, or proteins according to the molecular size. In gel electrophoresis, molecules can be separated or pushed by an electrical field through a gel that contains many small pores. The molecules travel through the pores in the gel at a speed that is inversely related to their length. This means that a small DNA molecule will travel a greater distance than that of a larger DNA molecule. Major uses for this include forensic science or diagnosing a disease. Sanger sequencing is a method used for determining the nucleotide sequence, base by base, of a fragment of DNA by the use of dideoxynucleotides, which cause the termination of DNA synthesis during this procedure.
PCR, polymerase chain reaction. It is an automated method used to amplify regions of our own DNA. We can refer to it as DNA replication outside the cell. And this is done by a series of steps before running the sample into a thermocycle machine or a PCR machine. However, before this can be done, we need to have samples of our DNA extracted.
Huntington disease. Huntington disease is an inherited neurodegenerative genetic disorder which is caused by an autosomal dominant mutation. This means that one affected copy of the gene can cause the disease. This gene is known as HTT gene and is also referred to as the IT15 gene, which resides in chromosome 4. Repeating units of CAG codons on chromosome 4 is responsible for the disease, and that is a trinucleotide repeat mutation. This mutation causes the formation of an affected protein. A Huntington disease patient usually starts to show symptoms of the disease at the age of 35 to 44, and the symptoms of the disease worsen throughout the span of 10 to 25 years. The disease affects the nervous system of the entire body, causing abnormal movements, cognitive problems, and psychological problems. Gene expression. Gene expression is divided into two stages, transcription and translation. For HD, the transcription of the DNA sequence that code for the Huntington protein is by forming an mRNA copy of the defective gene. The mRNA copy will be a replication of GUC codons. This mRNA will be modified before exiting the nucleus. It's modified by the addition of poly A tails, addition of 5' cap, and the splicing of non-coding regions by spliceosome. After the mRNA is modified, it is going to exit the cell, and this is where the second stage of gene expression will start. The defective gene will produce the Huntington protein by gene expression process in our cells. In translation, the mRNA sequence will get decoded in the ribosome by the tRNA sequence and decodons, which is a replication of CAG codons. Each CAG codon codes for the amino acid glutamine. A polypeptide chain of glutamate amino acids will be formed by the end of translation, and this polypeptide chain is the Huntington protein.
The symptoms may vary between each individual, but the main ones include undercognitive, sudden loss of recently learned skills, inability to react to others' feelings and emotions, loss of judgment, clumsiness, etc. For psychological disorders, there might be phases of depression or anxiety, or maybe even sudden irritability. Movement disorders, chorea or akinesia, fidgeting or tremors along with change in handwriting, and also experiencing difficulty in doing daily activities.
The Huntington disease affects the basal ganglia and the cortex. Striatum is the part of the basal ganglia that is most affected. It is made up of the caudate and the putamen. The motor cortex is the section which coordinates with the striatum, forming two pathways, direct and indirect. Huntington disease causes the death of nerve cells only even though the Huntington protein is active throughout the body. This interaction between the Huntington protein and two other proteins, Huntington's interactor protein and Huntington's associated protein, that is only present in the brain. Therefore, the defective Huntington protein affects only the brain. The defective Huntington protein have been known to bind to Huntington's associated protein, more causing of the death of striatal and cortical cells.
The direct pathway is disrupted when striatal cells die, causing the understimulation of the motor cortex. This causes the symptom of slow movements, whereas the indirect pathway, when disrupted, happens to involve the overstimulation causing chorea. In adults, the indirect pathway usually are affected first, which is the reason why chorea is seen as a primary symptom. In the case of juvenile Huntington's disease, both the pathways are affected simultaneously, which therefore eliminates chorea as the first symptom. There are no definite cure for the Huntington's disease. Doctors usually prescribe the patient medication to manage the symptoms for the disease. Dopamine is a neurotransmitter present in the striatum, therefore working against dopamine will actually relieve the activity of the nerves, thus stopping the chorea. Two examples of these dopamine antagonists that are used to combat against chorea are tetrabenazine and olanzapine. As for depression, hallucination and other mental disorders are common among Huntington disease patients. Antidepressant and antipsychotic drugs are regularly used to treat this. Through quantitative neurological examination (QUE), doctors are able to monitor the development of a HD patient's involuntary movements. Genetic testing is also able to test for the disease.
The protein structure of a defective Huntington gene will be vastly different from that of a normal non-defective Huntington gene. HD protein, since being defective, will go through protein aggregation, meaning that the protein will misfold and form clumps, which is correlated to Huntington's disease. Protein aggregates of Huntington disease is known to travel around in the cell, which means that the restriction of neural functions of the brain as protein aggregates is said to limit the nerve cell functions.